Hi, we are Team UCCKE from Hong Kong. In this video, we are going to briefly introduce our project. We were inspired by a YouTube video about a horsehair worm which infected a mantis. The infected mantis was drawn to water under the control of the horsehair worm. The mantis then proceeded to drown itself to return the worm to water. We wondered, if the horsehair worm could infect a mantis and control its behavior, could we do the same by biochemical methods too? We decided to try out this idea on the worm C. elegans. It's a kind of free-living round worm. It's easy to handle, and the cost of maintenance is relatively low. It is known that C. elegans attack the roots of our plants. The plants under attack would eventually die. So we plan to use our idea to keep these worms away from the crops. In essence, we will attempt to build a trap and fence system. For this, we ought to know what C. elegans likes and dislikes. So naturally, we resorted to Google to source out attractants and repellents for C. elegans. We came up with a possible attractant called cinnamaldehyde, which is a volatile organic compound. And we decided to use phenylpyruvic acid as the repellent. So now we have cinnamaldehyde and phenylpyruvic acid. All we need now is proof that they actually work on the worms. We designed a chemosensation assay to test whether the substances would attract or repel C. elegans, and thus determine how those substances would suit our project. For volatile substances, we will conduct a volatile test. As shown, water was loaded on the spots labeled C as a control, and the tested chemicals were loaded on T spots. Note that we are loading the chemicals on the lids of the dishes in volatile tests. We place the lid over the plate which contains C. elegans at its center. For the droplet test, we loaded the chemicals on different spots on the plate and placed C. elegans at the center. The worms are going to move to different areas in the next 30 minutes, and we are going to count the numbers then. The response index is calculated by T minus C over T plus C, where T is the total number of worms in the T areas and C is that in the C areas. So positive index indicates an attractant, while negative signs for a repellent. The index must lie between -1 and 1. Using the above assay, we found out a surprising result which contradicted our predictions. The cinnamaldehyde was in fact the repellent, and the phenylpyruvic acid was in fact the attractant. The response index of cinnamaldehyde was -0.83, which is quite strong, and the phenylpyruvic acid had an index of 0.34. So now let's move on to the cloning part. Generally, we designed our parts like this. It begins with a constitutive promoter J23100, followed by an RBS B0034. Here comes our gene. We will talk about this later. The parts end with a double terminator, B0015. Now is the time for us to find the sequences of our gene. Thanks to the 2013 British Columbia team, we obtained the sequences of the genes that were responsible for the cinnamaldehyde biosynthesis pathway. Including the 3 genes in the pathway, we cloned each gene into a plasmid and then cloned the 3 genes together. But for phenylpyruvic acid, how are we going to do it? It is a new addition to the iGEM community. On NCBI, we discovered a protein sequence enabling the production of phenylpyruvic acid. And here is our cloning map. So now we've got the gene sequences and the part design. After ordering, we received packages enclosing the DNA from IDT. We then cloned them into plasmids and then transformed our E. coli bacteria. Oops, I guess there's not enough space. Oh, and one more thing. We have to do the assays on the synthetic chemicals again to ensure our parts really do work as a biochemical attractant or repellent. And here are the results. We see that our synthetic phenylpyruvic acid is much more attractive than pure phenylpyruvic acid. That means we have succeeded in creating a new biochemical method of controlling other organisms. However, for cinnamaldehyde, we are limited by our high school lab and we are working hard to produce a synthetic form of it. Thank you for watching this video. We hope you enjoyed it. And we look forward to seeing you on Sunday, 3:30 p.m. at room 302 during our presentation. Thank you.